Thank you very much. It's a pleasure to be here with you. I'm going to talk to you a bit about the endocannabinoid system and the idea of the expanded endocannabinoid system that's being currently defined and its implications in cardiometabolic risk. This is the old, I guess there's not the new presentation, it's not up. I noticed the new presentation was downloaded, was on the website, but it's not on here. No, but I mean, I should be able to be okay. It's quite a bit longer, though, but I'll try and get through it. So we're going to have a brief introduction on the endocannabinoid system, its role in lipogenesis and lipolysis. Sorry, I'm worried about the animations not going the way I expect. Its association in obesity and its dysregulation in obesity, and then talk about the endocannabinoidome and its emerging role in cardiometabolic risk and atherosclerosis. So the cannabinoid receptor is the largest or the most abundantly expressed GPCR in the brain, but it's also expressed in a lot of peripheral tissues. Okay, sorry about this. And it's a unique GPCR that's not really, doesn't look like other GPCRs other than CB2, although their similarity is not that high. The reason it's called the cannabinoid receptor is because it was the original target for THC. And endocannabinoids are so named not because of their structural similarity to THC, but because of the fact that they activate the same receptors. Here's an example. It's 2-AG, 2-hydrocodonyl glycerol. It's produced from biosynthetic precursors found in the phospholipid bilayer, as is anandamide, AEA, which is a arachidonyl ethanolamide. So as I said, these are produced in response to metabolism of phospholipid-derived precursors by a variety of enzymes. NAPAPLD is the major enzyme for anandamide. DAG lipase is the major enzyme for arachidonyl glycerol. And these are subsequently degraded by other enzymes, hydrolases and lipases, that break them down to their component parts, which includes the production of arachidonic acid and other fatty acids that can be incorporated into this endocannabinoid system. So the endocannabinoid system is considered, when we talk about not just the ligands, but the receptors that are involved and the enzymes that are resulting in their production on demand and their degradation, again, on demand. So the ECB, or the endocannabinoid system, is considered a thrifty system. It's promoting the conservation of energy and the upregulation of storage of energy. And this happens not only in the central nervous system, but also in peripheral organs. As I said, and CB1 plays a main role here in all aspects of whole body energy metabolism. In the brain, it's regulating appetite and satisfaction to certain diets, as well as perception of fat and sugar content. It's also involved in the gut, to regulating hormone production and digestion and absorption, within the pancreas, regulating beta cell health and insulin production, within the adipose tissue, regulating inflammation that's happening there associated with obesity, but also regulating lipogenesis, adipogenesis, and glucose metabolism there. The same thing is happening in muscle, and it plays a key role in liver, where it's regulating, again, glucose metabolism that has an effect on whole body insulin sensitivity, as well as lipid metabolism. So this is just a picture of an adipocyte, and showing you the main points of function of CB1 activation within the adipocyte. It works not only at the endocannabinoids work not only at the cell surface, activating CB1, but also in the nucleus, activating nuclear targets, especially in the adipocytes PPARG, where it's regulating lipogenic and lipolytic transcriptional programs, as well as the adipogenic transcriptional program to upregulate the adipogenesis. It regulates glucose homeostasis, improving the amount of sugar that can get into the adipocytes. It's changing fatty acid synthases to regulate the production of de novo lipogenesis. And it's also acting at multiple levels on the mitochondria, where it's changing mitochondrial biogenesis, as well as beta oxidation and the ability to utilize fatty acids. In general, the picture is that overactivation of CB1 within the adipocyte and the adipose tissue is resulting in an increase in the amount of cells and the amount of lipids stored within the adipocytes. In the liver, we see a similar situation, an upregulation of lipogenesis and a modulation of glucose metabolism. Interestingly, you get in a non-alcoholic fatty liver disease, you get an upregulation of CB1 activity and endogenous arachidonyl ethanolamide production. The CB1 endogenous agonist, you can see it on the left-hand side there. But in an alcoholic fatty liver disease, you get an upregulation of the other classical endocannabinoid 2-AG, which is being produced by hepatic stellate cells, which is then going on to activate CB1 and resulting in similar changes. One of the main players that CB1 activation acts on is AMP kinase, which is a kinase responsible for the detection of energy levels within the cell, and this inhibition of AMP kinase is resulting in, again, an upregulation of the lipogenic pathway, a switch towards gluconeogenesis, and changes in, again, mitochondrial activity. There's also, the endocannabinoid system is also, by acting on the adipose tissue, regulating signaling from the adipose tissue to the liver. And if you look specifically within adipocytes, what we often see is that CB1 activation is upregulating ER stress, and this ER stress is resulting in changes in gene expression patterns, as well as the direct inhibition of insulin receptor substrate, which is changing the ability of insulin to regulate glucose transport into the cells, as well as modifying the ability of the liver to break down insulin by decreasing the amount of insulin-degrading enzyme, resulting in hyperinsulinemia throughout the whole body. And in cases where the CB1 has been shown to interact specifically within the hepatocyte and using hepatocyte-specific knockouts, it's present within the hepatocyte, actually results in whole body insulin desensitization. So in general, in obesity, the CB1 is overactive, and it's happening all over the place. It's happening in the central nervous system. It's happening in the adipose tissue to regulate the production of new adipocytes, as well as I said, the production of adipokines, the upregulation of inflammation, and then again in the liver to regulate lipogenesis and gluconeogenesis, as well as in muscle, which has an inhibitory effect on insulin activity in the muscle, and in the pancreas itself, where it's changing the ability of the pancreas to produce insulin. So as I said, the endocannabinoid system is dysregulated in obesity. This is diet-induced obesity in mice, showing you that the changes in the endocannabinoid tone at the level of the production of endocannabinoids themselves is different depending on whether you're talking about visceral fat or subcutaneous fat. And so you can see on the left-hand side in epididymal fat, which is a visceral fat, we don't see much of a reduction in AAA in this model, but 2-AG levels are upregulated significantly. In the subcutaneous fat, we see the opposite effect. Anandamide is decreasing, as is 2-AG. Okay, this is from human studies in obese patients, and you can see 2-AG there in the middle in visceral fat. Again, it's upregulated, and there's a slight downregulation in the subcutaneous adipose tissue. And when we look at CB1 receptor expression within the visceral adipose tissue, we see that that's actually increased as well, indicating the tone at the level of both the endocannabinoids and the receptor is upregulated here. This is data coming from obese patients with type 2 diabetes, and their changes in the endocannabinoid system with respect to pre and post bariatric surgery. So on the left-hand side, you can see lean patients, subcutaneous and visceral adipose tissue. You see that, excuse me, CB1 expression levels are decreased in subcutaneous adipose tissue, but increased in visceral adipose tissue. And I think I got rid of this data in the subcutaneous adipose tissue as well. On the right-hand side, this is in patients that are going to be undergoing bariatric surgery. You see this decrease as well. And I got rid of this slide as well. And 2-AG again is downregulated here. And these are the changes in the endocannabinoid system pre and post bariatric surgery. You can see in subcutaneous tissue, you can see that AAA is now increasing significantly, and 2-AG even more. So bariatric surgery resulting in significant amounts of weight loss, correction of dyslipidemia, it also results in a significant upregulation of the endocannabinoid system within the subcutaneous adipose tissue. So put this data together, and we think that what's happening is that this disbalance in the endocannabinoid system and the differences between the subcutaneous tissue, adipose tissue, where it's decreased, and the visceral adipose tissue results in differential responses in obesity. You have less accumulation in the subcutaneous adipose tissue as a result of this, and more accumulation in the visceral adipose tissue, which is, of course, metabolically unfavorable and associated with poor response to insulin, dysregulation of glucose levels, and atherogenesis as well. So 2-AG itself, circulating 2-AG levels, is associated with several risk factors for cardiometabolic disease, including body weight, waist circumference, and intra-abdominal obesity. It's negatively correlated with HDL, but it's positively correlated with fasting insulin levels, triglycerides, as well as glucose. So patients that were put on corrective lifestyle change that include changes in diet as well as physical activity were assessed for endocannabinoid levels, circulating endocannabinoid levels, and you can see here that both anandamide and more specifically 2-AG, more strong 2-AG levels are decreased in this case. And these decreases in 2-AG were positively correlated with decreases in visceral adipose tissue, as we would expect, decreases in free triglycerides and HDL and LDL, increase in HDL cholesterol, and a decrease in insulin resistance as well. So CB1, because of the intimate regulation in whole body energy metabolism, was targeted, of course, as treatment for obesity, and the drug that was on the market was known as rimonabant, and this is showing you the effects of rimonabant on visceral and subcutaneous adipose tissue, and we can see that rimonabant has a much stronger effect on the visceral tissue than on the subcutaneous adipose tissue, which is in line with what we see with respect to the overactivation of the system within the visceral tissue itself. As well, rimonabant was associated with weight loss independent effects on rimonabant on various parameters of cardiometabolic risk, and a certain amount of this was